Okay, it's noon. I think we're ready to get started. So good afternoon to everyone who's joining us. My name is Mark Freeman, and I'm the director of the Volum Institute at OHSU here in Portland, Oregon. The Volum Institute is a privately endowed research institute on OHSU's campus that is dedicated to basic research in neuroscience, and the hope is that our work will lead to better understandings and new treatments for neurological and neuropsychiatric diseases. I'm delighted to be here today to kick off the OHSU Brain Institute's Brain Awareness Lecture Series. Welcome to our viewers from around the country. I have first a few housekeeping items I need to announce. We welcome your questions and look forward to them. Please email Kate Stout at any time now throughout the talk, and we will relay these questions to Dr. Monk at the end. I'll read them to her in a Q&A session. Kate's email is Stout, STOUTK, at ohsu.edu. You can also find her email in your registration confirmation. If you're having any technical issues, you can also email Kate for troubleshooting help. The lecture is also being recorded and will be posted to the Brain Institute's website. So if you'd like to, watch it over and over, you're welcome to do that. It will be there and you will have the links. Finally, I want to thank you all for your support of the OHSU Brain Institute. Your philanthropic support is really critical, especially now, to moving basic research forward from the bench to the bedside of all those patients in need. So now I will take some time to introduce Dr. Monk. And that also reminds me I need to shut my phone off. So it's my great pleasure to introduce Dr. Kelly Monk. Dr. Monk is a professor and the co-director of the Volum Institute and the director of OHSU's Neuroscience Graduate Program. So she oversees our major program in neuroscience that trains graduate students. Dr. Monk received her Ph.D. in cell biology in 2006 at the University of Cincinnati, working with Dr. Nancy Ratner. There she studied ways in which peripheral nerves, the nerves that connect our brain and spinal cord to the rest of our body, respond to injury or tumor growth. She did some really beautiful work in that area, and this began her interest in trying to understand how nerves in the nervous system are built, how they function in our bodies, and how they change in disease and lead to functional loss in patients. The primary model that she used in the laboratory at that time was mouse, which was great and really allowed her to get her foot in the door, but it had some limitations. And she then moved to Stanford University and did her postdoctoral work after her PhD with Dr. Will Talbot. And one of the really important things Dr. Monk did was he helped the Talbot lab pioneer the use of zebrafish to study the biology of myelin. Myelin is, probably many of you have heard of it, it's that fatty sheath that surrounds neurons, especially the long fibers, the axons. And it allows neurons to convey signals at remarkably fast rates across the nervous system. And you'll hear much more about that from Kelly during her talk. It is also the structure that breaks down in really devastating diseases like multiple sclerosis, and myelin loss is a common feature of many neurodegenerative diseases. Her decision to exploit the powerful tools available in zebrafish to study myelin was really astute, in my opinion, I think in many people's opinions, and it's paid off tremendously. Myelin had been studied for decades, but when Kelly started to use the zebrafish, we didn't really understand how you build myelin. And understanding how to build myelin or make new myelin in the nervous system is really the central goal for the field, because having the ability to do that is key to treating patients with diseases like MS. Impressively, Dr. Monk discovered what others, real luminaries in the field, referred to as the holy grail for the field, and that is, as you'll hear, a receptor called GPR126. It doesn't really matter what it's called. What it is a molecule that is, it seems, the key regulator that drives the formation of myelin in many parts of the body. So there's actually the potential to go in there and actually start regulating when myelin gets formed. Dr. Monk moved to Washington University in St. Louis in 2011, where her lab performed many of the key studies that lead to our current understanding of GPR126 and how it regulates myelin formation. Her group also made many additional discoveries in myelin biology that I'm not going to list, but by all measures, her laboratory was exceptionally successful. We were lucky enough to recruit her to the Volum Institute at OHSU in 2017 where she was appointed the co-director of the Volum Institute and the following year director of the neuroscience grad program. Dr. Monk is recognized as a worldwide leader in the study of myelin biology. She serves on many distinguished advisory panels on the topic and was recently, in fact, named chair of the NIH study section on the cell and molecular biology of glia. She's received numerous prestigious awards for her research, and her work has been supported by many grants from the National Institutes of Health and the National Multiple Sclerosis Society. Since joining the Volum Institute, she's continued her amazing work on myelin. You're going to hear more about that today, and she's branched out into many other interesting new areas in neuroscience, in particular with a focus on how cells of the brain that are not neurons, called glia, which are about half the cells in the brain, help build brains, help drive their function, and keep the nervous system healthy. Finally, I just want to point out Dr. Monk is also well known as an outstanding and unusually attentive mentor, for which she's also received quite a bit of recognition. So in addition to doing amazing science, she's helping us train the next generation of leaders in science. So with that, I want to say thank you to you all again for coming, and I'll hand it over to Dr. Monk.
Thanks a lot, Mark, for the really kind introduction. I'm super excited to be kicking off this lecture series and to tell you about the work that my lab has been doing over the years and currently using zebrafish as a tool to understand how the nervous system functions. And so if I asked any of you listening to this lecture to think of a cell in the nervous system, I think it is fair for me to assume that almost everyone would think about a neuron, maybe a particular type of neuron, or maybe just neuron in general. That's reasonable. That's a completely rational thing for you to be thinking about. Neurons, of course, are the basic functional unit of the nervous system, the most fundamental processing unit in our brains. And the study of neurons has been really, obviously, the focus of neuroscience for well over 100 years. Now, if you take anything at all from this lecture at the end of it, what I really hope you'll remember going forward from today is that neurons are not alone in the nervous system. As Mark mentioned in his introduction, neurons are actually only about half of your brain, and the other half is comprised of this really diverse and remarkably interesting population of cells collectively known as glia. There are many different types of glia. I don't have time to get into all of them. I'll just point out some of the more famous ones here in this cartoon. Microglia are the resident immune cells of the central nervous system, the brain and spinal cord. They respond to pathogens and injury. They're really important in disease states, and they're really important as well in sculpting the nervous system during development. There are these star-shaped cells called astrocytes, shown here in this cartoon, that are really intimately associated with neurons, as well as with blood vessels, with the vasculature. And so astrocytes are really important for regulating neuronal function, as well as regulating blood flow and providing oxygen and glucose to the brains in areas that really need it. Finally, a third sort of famous type of glial cell is the oligodendrocyte in the brain and spinal cord. And these are the cells that make myelin. And I'm going to come back to oligodendrocytes and myelin in just a moment. But I want to just make the broader point about glia. So the word glia derives from a Greek word that means glue. And it was, these cells were given this name back in the 1800s because it was thought by neuroscientists at the time that all these cells really did was function as sort of like cement or really glue, holding the rest of the nervous system together, and really only functioning in supportive or structural roles. We know that is categorically false. Glial cells provide numerous essential functions in the brain, in development, in normal health, and in disease states and after injury. And we're really just beginning to scratch the surface of what these remarkably interesting cells do. My lab and I are interested in all these different types of glial cells in the nervous system, but what I do want to focus on today is work that we've been doing to understand myelination in both health and disease. And so, as I mentioned, the cells that make myelin in the central nervous system are called oligodendrocytes. What is myelin? I just want to briefly introduce you to what this structure is. You've all probably heard of white matter. white matter is called white matter because of myelin. So myelin is this lipid-rich, this fatty membrane that's created by cells. This membrane wraps around projections of a neuron called an axon, and at the end you have this insulation, very much like my earphones are insulated by plastic, the wires underneath. The axons are insulated by myelin. It's this fatty, lipid-rich, white appearing substance. And so it's made by cells called oligodendrocytes in the central nervous system, the brain and spinal cord, and by Schwann cells in the peripheral nervous system. I may use these different names interchangeably, or I may just refer to them as myelinating glia throughout the course of this lecture. But what myelin does is insulating these axons, it allows for really fast communication between neurons. There are actually two ways that you can make neurons communicate quickly. One is to make axons really, really big, really large diameter. And this is what certain invertebrate species like squid or octopus have done. They have a really large diameter axon. That works well if your nervous system is not encased in bone. But if your nervous system is encased in bone, it's very difficult to have large diameter axons. So I think it's really fascinating that we know that myelin actually evolved in the vertebrate lineage when jaws evolved. So you've probably heard about, what lampreys are. There are these jaw-less vertebrates. So lampreys do not have myelin, but all vertebrates with jaws do have myelin. And so myelin is thought to have arisen actually in the Devonian period, about 380 million years ago, with the acquisition of jaws. So why did myelin co-evolve when jaws evolved? Well, imagine you are a fish now with jaws and you want to hunt your prey. You need to be fast to catch it. Or imagine you're a fish that's being hunted. You want to be fast and swim away from your predator. And so what myelin did over the course of evolution was to allow for, really important in the evolutionary success of the vertebrate lineage, because it allowed for a really complex nervous system that's encased in bone to evolve. It's been estimated that if humans did not have myelin, but we were still as fast and as smart, if we were still functioning exactly how we function, but that we had evolved large axons instead of myelin, our spinal cords would actually need to be the diameter of a giant sequoia tree. And so this acquisition of myelin in the jawed vertebrate lineage was really critical for the success of vertebrates over the course of evolution. So I think that's fascinating, just in terms of the evolutionary history of myelin. But when you think about myelination today in humans, it remains fascinating. So myelination occurs almost entirely after birth. This is one of the longest protracted developmental processes that occurs in our nervous system. We are all actually normally myelinating, developmentally myelinating, well into the 4th decade of life. What you're looking at here are sections of the human brain. This is from a paper from about 100 years ago. And these brain sections are stained with a histological dye in which myelin is marked in blue. It's actually labeling the fat. So this is the white matter shown here in blue. And so here you can see at birth, there's very little myelin in the brain of this baby, but over time, as humans develop and grow, the myelination throughout the brain really expands throughout the entire white matter. Moreover, beyond this developmental myelination, we know that it's actually quite plastic. So if you learn a new skill, for example, learning to play a musical instrument or learning to juggle, we know from imaging the brain before and after acquiring this new skill, the myelination patterns in the brain can actually change in response to acquiring a new motor skill. And so how this expansion of myelination in development or in response to experiences is impacting neural circuits, learning, and behavior is a really fascinating question for the field to ponder. Beyond these big picture questions about circuits and behavior, I think it's fair to say that there are a lot of outstanding cell biological questions that are really fascinating for the field. And these are some of the questions in particular that my lab is interested in, trying to understand how myelin develops. So as I mentioned, what myelin is this extension of the membrane from these myelinating glial cells. The membrane wraps many times around the axon to ultimately make the myelin sheath. How do the cells know which axons they need to myelinate? How do they know how much myelin to make is a really interesting question. I'm showing you here an image of two different oligodendrocytes in the spinal cord. This is the cell body or the nucleus of these oligodendrocytes, and these are the processes spreading out from the center of the cell. The myelin is marked here in white. This is a fluorescent marker of myelin. And all these little tubes that you see, these little tubular segments, these are the myelin sheaths forming. The axons are present, you just can't see them because they're not labeled. But I think it's really interesting to look at these two oligodendrocytes in the spinal cord, and you can pretty readily observe that this oligodendrocyte is making many myelin segments, but they're all a bit short compared to this oligodendrocyte, which is making fewer myelin segments, but they're quite a bit longer. How do these cells know how much myelin the axons need? How do they know how long the myelin should be, how thick the myelin should be? These are really interesting fundamental questions that we're excited about. Additionally, how do these cells know what they should and should not myelinate? Now, in the brain, in the spinal cord, we know that more active neurons are more likely to have their axons myelinated. However, if you take these oligodendrocytes out of the brain or spinal cord and culture them in a dish, and instead of culturing them on axons, you can actually culture them on completely inert structures. So in this example, the oligodendrocytes have been cultured on polystyrene nanofibers. So that's dead. There's no firing, there's no neural activity going on in a polystyrene nanofiber. But what you can see here in green is that these oligodendrocytes have made myelin around these nanofibers, shown here at the light level, and this is shown myelin at the using a microscope called an electron microscope, which allows for really high resolution imaging. And all these little lines are the individual wraps of myelin, instead of around an axon, around a nanofiber. So what this means, and I should point out that the oligodendrocytes will myelinate these nanofibers provided they're a big enough diameter. So what that means is that the oligodendrocyte has this intrinsic capability of making myelin around a tube as long as that tube is the proper size. However, in the brain, there are lots of structures that are the right size and diameter to be myelinated, like blood vessels, like other processes of neurons or other cell types. But we know that those structures do not get myelinated in the brain. And so there must be inhibitory and myelin promoting cues that the oligodendrocyte has to process in order to output myelin at the right spot, at the right time, the right length, at the right thickness. So really interesting to think about. Now, there are two different types of glial cells that make myelin, the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. And they're thought to have arisen at the same time in evolution. But the way that these cells make myelin, although myelin serves the same function in the CNS as in the PNS, the way that these cells make myelin is actually quite different. An oligodendrocyte is capable of making myelin around dozens of axons, whereas a Schwann cell only makes myelin around a single axonal segment. That's interesting to think about why these cells evolved such fundamental differences in how they myelinate, but that also might be really important in the context of disease, injury, and repair, because the peripheral nervous system is relatively better at regenerating compared to the central nervous system. And maybe part of that has to do with these fundamental differences between the myelinating glial cells of the peripheral versus the central nervous system. We also, as a field, I think, logically have focused on functional interactions between neurons and glia. And this is, of course, very important to understand how the brain works. But in this really busy cartoon, what you can see is that not only are neurons not alone in the nervous system, but glia are not alone in the nervous system. This is half of your brain. How are they interacting not only with neurons, but also how do these glial cells interact with other glial cells? How does this oligodendrocyte interact with this microglial cell or this astrocyte? And do these interactions influence myelination in development or in the disease state? And so, we can talk about how interesting myelin is from an evolutionary perspective, or how fascinating the cell biology is of these cells that make myelin. But the importance of myelin, I think, is really underscored in a disease like multiple sclerosis, where the body's immune system is attacking the myelin sheath. I'm pretty sure that everyone listening to this lecture knows at least one person that's affected by MS. It's a very common disease in the United States alone. Million people suffer from the disease, and in fact, in the Pacific Northwest, the incidence is quite a lot higher than in the rest of the country. And we can infer the importance of myelin in the properly functioning nervous system, because when myelin is lost or damaged in a disease like MS, this really underlies and contributes to many of the debilitating symptoms and can lead to neuronal loss that's observed in this disease and other diseases of myelin. And so what we really want to get at by understanding, the basic biology of the cells that make myelin, what we really want to get at is to understand how we might promote remyelination, how we might promote repair in a diseased or injured brain. And so to get at that, I think a couple of key questions are, when myelin is formed in development, during normal development, is this a different pathway perhaps than what might occur in the repair state? For example, in MS, the cellular environment that these cells are surrounded by may be quite different in the developing brain versus a brain with MS. And we also wonder, even if these pathways are different between development and repair, if we fully understood the development of these cells, might we be able to harness these pathways that are required in development to promote repair in a myelin disease or after injury? And so one of the tools that we use in the laboratory to study myelination and to study the nervous system in general is this model organism shown in this image called a zebrafish. And so why do we use zebrafish? So if you are, why did zebrafish even become a model system that we use in the laboratory? So if you have an aquarium at your house, or if you've ever been to a pet store, you probably recognize these fish. They're either called zebrafish, zebra danio, or danio rario. They're really cheap. They're like the cheapest fish in the pet store, honestly. And the reason that they are so inexpensive is because they're really easy to care for. These are great starter fish when you're just getting into aquaria. They do really well in a home aquarium or in the laboratory aquarium. And they breed, accordingly, they breed very well in the laboratory. And that made them a really excellent model system to be developed. And I think it's really interesting to note that zebrafish research, while today there are thousands of labs around the world using zebrafish as a model system for many really important biological questions, the whole field of zebrafish research and studying zebrafish in biology began right here in Oregon, down in Eugene at the University of Oregon, pioneered by a professor named George Streisinger, and he established zebrafish as a model system in the 60s, the 70s, and the 80s. Eugene, U of O, and Oregon in general are really a major hub of zebrafish research in the world. Is one of the reasons I was really excited to move my laboratory here. And it's really taken off around the world. Of course, I'm biased, but I do think zebrafish really represents one of the most exciting systems that we can use in contemporary neuroscience to understand how the brain works. And so here at OHSU, in my laboratory, we built a brand new zebrafish facility to house our zebrafish and also collaborators. We have the capacity to house about 40,000 zebrafish. We have 1,800 tanks in our facility. And kind of excitingly, we actually have a robot that feeds the fish. And I'll show you this robot in action here. This robot can read the barcode that's on all of the tanks and distribute food to the tanks according to the number of fish that are in there. That sounds kind of simple, but what that robot actually does is allows our fish to grow very, very rapidly. So zebrafish grow as a consequence of how much food they're given. It's hard for humans to feed more than twice a day, just for logistical reasons, but the robot can feed 8, 9, 10 times a day. And what this allows allows our research to go very, very quickly, and for our fish to reach maturity in about half the time as a lab without this robotic feeding capability. When the pandemic is over, we're really happy to have to provide tours of the fish facility. If anyone would be interested, definitely just reach out, and we're happy to show you the facility, to show you the fish, to show you the fish under the microscope, and to show you the robot in action. Okay, so why do we use fish to study myelination? So as I mentioned at the beginning of the talk, myelin is this evolutionary innovation of the jawed vertebrate lineage. And so what this means for us in practice is that zebrafish is the simplest lab animal that we can use to study myelination. And we hope that by making fundamental discoveries in zebrafish to find conservation of these genes, of these molecules, by understanding it in the simple possible first, we can then ramp up into studying humans. Zebrafish, so in the adult, those beautiful stripes that when they're larvae, when they're still developing, the pigment isn't fully developed yet, and so we can actually see into the nervous system of these fish in development. And so additionally, because we can get many zebrafish embryos from a single pair of parents, these animals are amenable to large-scale genetic and chemical screening. And I'll go through all of these points in the coming slides. Importantly for us, zebrafish myelin is very similar to myelin that's found in mammals, in mouse or in human. These are two cross sections of nerves using that electron microscope that allows for really high resolution imaging. The black rings are myelin sheaths surrounding these white circles, which are axons. One of these samples is from a mouse, and one of these samples is from a fish. You can see that they're really quite similar. This happens to be the mouse, this happens to be the zebrafish, but what I think this underscores is how fundamentally similar myelin is in zebrafish compared to mammals. And so the discoveries we make in zebrafish, we can scale up and study in mammals. I'll show you a couple of movies that I think really highlight the power of zebrafish as a model system. The first is that these animals develop very quickly and they develop externally, and so we can watch development happen. Of course, mammals develop inside of the mother. It takes a very long time, and we can't observe development in real time. And what you're going to see here is a movie of the first 18.5 hours in the life of the zebrafish embryo. When the movie starts, the animal is about 45 minutes old. Down here is yolk, and on top of the yolk are two cells. This was a single cell that's divided into two. And what you'll see, this movie is sped up, but the first thing that happens in the development of this embryo are lateral cell divisions on top of the yolk. After about 3 hours, this is a blastula of 1,000 cells. What you're going to observe are these cells migrating over the yolk. Very soon, a head is going to form. You'll see eyes forming, muscle segments forming, neurons are being born, the axons are extending. A few hours later, glia will be born and start to populate the nervous system. This happens very, very, very rapidly. That was less than 24 hours. And you already have an animal at the end of it that looks a lot like a zebrafish. And in just a few days, the nervous system will be very complex, and the animal will be performing very complex behaviors. with a more high-powered microscope, we can actually watch in real time the cells that make myelin directly in zebrafish. And so I'm going to show you a couple of movies taken in my laboratory from the spinal cord of a zebrafish. And the cells in black are different oligodendrocyte cells at different stages of development. In this movie, we're looking at oligodendrocyte progenitor cells, or OPCs. Shown here, this is the cell body of this OPC, and all of these processes is the membrane extension of this oligodendrocyte progenitor cell. It's feeling around, it's trying to decide what it should myelinate, what it should not myelinate. Axons are present but not visible. Other glial cells are present but not visible because we're only looking at single cell resolution here. But you can see how dynamic and active this cell is as it's developing and surveying its environment. Now, just a little bit later in development, this OPC will transition into a myelinating oligodendrocyte, and that's shown here. So by the end of this movie, you'll see these little black tubes, and those are the nascent, newly formed myelin sheaths. So in this movie, in a living animal with no perturbations, what you're looking at is the birth of an oligodendrocyte from its progenitor cell to a mature myelinating oligodendrocyte. This type of imaging is really just impossible in other model systems. So zebrafish allows us to make fundamental discoveries of molecules that are conserved in mammals, but it also allows us to just look at the nervous system in real time and see what's going on and really watch how these cells perform in development or when we perturb them in various ways. And I'm going to talk to you about how we might perturb the system here. And we do all of these things because we not only want to understand how the brain develops and how it functions, but we really want to understand how we might stop progression in diseases of myelin, like MS. And once the progression is stopped, how can we repair the damage caused by MS? The way that my lab has approached this is to try to understand the fundamental biology of the cells that make myelin in development and health. Because you may be aware in human MS lesions, there are actually a lot of oligodendrocyte progenitor cells sitting there, but they're not making myelin. They're not doing their job. And so it's also really important to note that all of the therapies available for patients with MS on the market today target the immune system. And that's really important. It's really important to dampen down the immune system in an autoimmune disease. But what we also need to do is figure out how to coax these cells that are there into making myelin in these lesions in the MS nervous system. And so we try to understand, how does an oligodendrocyte progenitor cell become a myelinating oligodendrocyte? What are all of the processes that occur in development to make functional, mature myelin? And so what I'm going to tell you about is how we've used zebrafish over the years, particularly zebrafish genetics, to uncover a new regulator of myelination. Talk to you a bit about the utility of zebrafish in chemical screening and how we can use this for pretty high throughput drug discovery. And then end on what I think is really the promise of zebrafish for personalized or precision medicine. And so I'm going to spend the next 10 slides or so talking to you about some data that we've acquired over the years. This may get a bit jargony at times, and so for the non-scientists on the lecture, if anything's not clear, as Mark pointed out in the introduction, we'll have a Q&A session at the end. Happy to clarify anything. So one great strength of the zebrafish is this ability to do genetic screening. And so we can find mutations that affect any process of interest. In our case, we're interested in myelination. One way that we can do that in zebrafish that's very, very difficult to do in mammalian systems is what's called a forward genetic screen. You may have heard of CRISPR-Cas9, CRISPR technology in the news. That's a way to break DNA exactly where you want to target the DNA. That would be reverse genetics. But what this is here is it's called a forward genetic screen. And so basically, what we want to do is break different molecules that are important for myelination, for nervous system development, in an unbiased and random way, and then go back and figure out what genes did we hit, what mutations did we make in the genome. And to do that, what we do is we take a chemical mutagen called ENU, and this introduces point mutations, so single base pair mutations at random throughout the genome. And this fish will have many, many different mutations. We breed these mutagenized fish over multiple generations, and we actually screen the great-grandchildren of the originally mutagenized fish. And what we're looking for are mutations that have been driven to homozygosity, so 2 copies of the mutated gene. And we're looking for zebrafish embryos that present, in our case, with disrupted myelin. You can screen for anything you're interested in. In our case, we were screening for disrupted myelin. And so here's what the fish look like that we were screening for. This is a 5-day-old fish. The head is over here, the tail's cut off. But again, where you see the white signal, this is myelin in both the central and the peripheral nervous systems. And here you can see the same myelin label in green. MBP stands for myelin basic protein. This is a structural component of the myelin sheath. And what we found in this screen are 2 independent mutations in a gene called GPR126. One was a premature stop codon, a strong loss of function mutation, and the other was a missense mutation. And what you can see, so compare this to the wild type animal that has lots of green signal. In this mutant, there's absolutely no myelin at all along this nerve. And in the weaker mutant, this hypomorphic mutant, we have reduced but not absent myelin. And I'm going to come back to that mutant at the end of the lecture, because it's a really important tool for us to understand how this gene functions, how GPR126 functions. Okay, and so we did a lot of work to understand how GPR126 works. And one of the experiments we did was to look with this electron microscope, and what we found is that when GPR126 is mutated, the myelinating glia can surround the axons, but they never wrap their membranes to make myelin, so they get stuck at this promyelinating stage. That's shown here from a zebrafish nerve. The axon is this is a cross-section through the nerve. The axon is in yellow, and the mutant Schwann cell is in purple. Here in wild type, the green labels really nice myelin that's been made. Now, importantly, we knocked the same gene out in mouse and showed that when GPR126 is missing from the mouse myelinating glia, we also get this arrest at the promyelinating stage. So we discovered the gene in zebrafish, but then found that it is essential for myelination in mouse as well as in zebrafish. So what is this molecule? It's a G protein coupled receptor. And what G protein coupled receptors are they are proteins that sit in cell membranes, and they convert signals from outside of the cell to inside of the cell. So this is the part of the molecule that sits in the cell, and GPR126 belongs to a subfamily of G protein coupled receptors called adhesion G protein coupled receptors. So they have a very long extracellular segment and this standard GPCR segment. But what's really important is that to note is that GPCRs in general are excellent drug targets, because you can target them from outside of the cell and then cause intracellular signaling events to occur. Something like 2/3 of all available pharmaceuticals on the market today actually target G protein coupled receptors. And so what we found through a lot of work in zebrafish, in mouse, and in the dish is that GPR126 sits in the myelinating glial cell membrane, receives signals from outside of the cell, initiates downstream signaling, specifically cyclic AMP elevation, turns on myelin genes, and myelination begins. We also did a lot of work to show that exactly how these receptors are activated is really quite fascinating. And so the protein, when it's being processed, is actually cleaved in half, although it stays together, traffics to the cell membrane. There's this cleavage event that generates this little fragment here, this red square, called what was called the Stockel sequence. And so this is a German word for stinger, because this fragment of the receptor actually activates itself. And so like a stinger, the Stockel flops down onto the receptor and activates it. This discovery was made in a dish, and we could tell from the signaling events downstream of the molecule that this is what was happening, that this Stockel was required for activation. But what happens in a dish is not always the same as what happens in vivo. And so we wanted to use the power of the fish to prove that this Stockel signaling was driving myelination. And so to do that, we now returned to reverse genetics, in which we can engineer precise mutations in the genome of these animals. And so what we did was to make a very precise 2 amino acid deletion in GPR126. So the whole protein, the whole molecule is present, but there's just 2 important amino acids missing from the Stockel domain. And what we found when we engineered this mutant is that the receptor could not function. And so again, looking at electron micrographs of the nerve, the green axons in wild type are surrounded by the black myelin rings. But when we specifically mutate, precisely engineer the Stockel sequence mutation, the myelinating glia are not capable of making myelin. Now, importantly, we could also show that the Stockel sequence is sufficient for myelination. So because the fish live in the water, we can actually take compounds, add them to the water, and see what happens. So our collaborators synthesized this Stockel peptide. We added it to the fish water, and we found that this could rescue myelination, and that is shown here. So now myelin is shown in purple. You can see nice myelin in the wild type animals, either treated with control or with the Stockel peptide. Here in the GPR126 mutants, we have no myelin, but if we treat with the Stockel peptide, we can rescue myelination in these mutants. So this is really exciting. This shows that this receptor can be targeted by a compound to stimulate myelination, not only in a dish, but in a living animal. So we're really excited about the prospect of GPR126 and engineering these Stockel peptides perhaps for drug discovery and promoting myelination in vivo. We've done a lot of work in my lab and with collaborators around the world to understand how GPR126 functions, as well as how other related adhesion GPCRs function in myelin development, in nervous system maintenance and repair in both the central and the peripheral nervous system. We've shown that the way that this Stockel peptide is exposed is actually through mechanical activation, and that seems to be the case for the family of adhesion GPCRs. And we're also starting to work with collaborators to figure out the unique structures of these molecules using crystallography. And so I point you to these papers that have come out over the years if you want to learn more about GPR126. But I hold this up as an example of the power of zebrafish genetics in allowing us to find completely novel regulators of myelination. So we found GPR126 mutants in a genetic screen in zebrafish. This wasn't hypothesis driven. This was purely discovery based. We were looking in an unbiased way for genes that affect myelination. And we found that in these mutants, the myelinating glia are stuck. It's like GPR126 is a switch that turns myelin on in these cells. And so in zebrafish, as in mouse, the glia cannot make myelin when this receptor is mutated. Now, really importantly, a few years after our work came out, human patients were found with human patients with a disease called arthrogryposis multiforme complexa were born, and these patients don't bend their limbs at the joints. Unfortunately, these babies passed away a few hours after being born, and they were sequenced. And what the physicians found is that the mutation in these patients with arthrogryposis multiforme complexa was mutation in GPR126. And so the physicians had saved the nervous system tissue of these patients. They went and looked once they knew what the gene was, and they realized that these human patients are also not making myelin in development. And so this is a nerve from a control sample. The burgundy color is showing you myelin. These are human patients with a GPR126 mutation, and there's absolutely no myelin in these nerves. And so the genetics of zebrafish really allowed us to discover this receptor that is conserved in humans, and it also highlights the utility of targeting this receptor for myelin therapies because the receptor is functioning the same way in humans. So how can we get at targeting this receptor? In addition to genetic screens, both forward and reverse genetics in zebrafish, zebrafish are also really the premier vertebrate model system for chemical screening for drug discovery. The fish live in the water, and so we can simply add drugs and see what happens, like I showed you for the Stockel peptide. So what we're doing in the lab currently is a high throughput screening platform to identify both modulators of GPR126 as well as myelination in general. And so to do this, we acquired a VAST system, which stands for Vertebrate Automated Sorting Technology. We array the fish, 3 fish per well in a 96-well plate, and a robot can suck the fish out of the well, send them through this tubing system into a microscope, image the fish. So the fish go into a capillary tube, they're spun around, and we can image myelination in these fish in response to different compounds that have been added. Now, importantly, we can set this robot up before we go home for the night and let the robot do screening of different compounds, come back in the morning and see what happened to the myelin in these animals. With regard to GPR126, we're taking that hypomorphic allele that I told you about. So this mutant has reduced but not absent myelin. And so with this genetic tool, with this mutant, what we're able to find are compounds that can rescue myelination, and we hope that these might represent good targets, good candidate therapeutic targets for myelin repair in humans. And we might also find compounds that reduce myelination further in these mutants, and this might tell us something about how GPR126 is functioning, or give us new insight into myelination in general. And so I hope I've conveyed to you the many strengths of zebrafish, using zebrafish as a model system to study brain development, to study myelination. I've given you one example, just one example, and that's the example of GPR126, how we've gone from discovery of this novel molecule, showed conservation in mammals, found out later that the protein is conserved in humans, dove deep into the mechanism, figuring out how this receptor functions, what's upstream, what's downstream, and how we can target it using drug discovery for promoting myelin repair. But that's just one mutant that's come out of the screen. We've done other screens in the lab, and we actually have 30 mutants sitting there waiting to be analyzed. I'm showing you just 2 mutants here. So you're looking down at the fish head. These are the eyes. Again, the purple is myelin. This is how myelin normally looks when we screen for it in the brain and spinal cord and the peripheral nervous system. You can see that this mutant has no myelin at all, and this mutant has increased myelin. So we're really interested in following these up, as well as many other mutants in the screen, any one of which might represent the next GPR126. I told you today about myelinating glia, and myelinating glia are very important in the proper nervous system functioning. But just like neurons aren't alone, obviously myelinating glia are also not alone. Zebrafish possess all the glia that are all the major glial classes that are found in humans, including microglia and astrocytes, and we're very interested in my lab in all the major glial classes, understanding how they develop, how they interact with each other. So what I've shown you for myelinating glia and GPR126, we can extrapolate this and discover for all the other classes of glia. And then finally, I just want to end on what I think is really the promise of zebrafish for precision medicine. So like I showed you, we can engineer precise mutations in the DNA of zebrafish using CRISPR technology. And so what we can do with this, thinking about humans, is, you know, if there's a human patient found with a certain mutation in a certain gene and it's unclear if that mutation is causing the disease, or if it is clear that the mutation is causing the disease, but it's not certain how it's causing the disease. As long as that stretch of DNA is conserved in fish, we can go in and basically make a humanized zebrafish, take the precise mutation that's found in humans, make that mutation in zebrafish, and see what that mutation does to the development of the nervous system, but also do drug screens on this humanized fish, really precision medicine, really personalized medicine for individual patient mutations. And this is compared to mouse, extremely rapid and quite relatively easy to do in zebrafish. And so with that, I wish I could tell you about everybody's project in the lab. I have an absolutely wonderful lab. Mark very nicely mentioned in the introduction about mentoring, but it's really easy to be a good mentor with such fantastic people in the lab. So I have a great lab currently. Many previous lab members have contributed to the work that I showed you, collaborators around the world, great collaborators here at OHSU, and our funding sources over the years. With that, I think we're gonna open it up to the Q&A now. And so I'm really excited to take your questions, and if we don't get to everything, please feel free to email Kate or myself.
All right, thank you very much, Kelly. I'm clapping for everyone for you. Well done, excellent. I have a number of questions here, and I guess we'll start with some questions about GPR126. So one of the first questions from the audience was, how do you go about deciding which compounds to test for effects on GPR126 in your screens?
That's a great question. You could think about doing this a number of different ways, and I don't think any way is right or wrong. What we decided to do, and we've done actually 2 different drug screens for GPR126. The first was a really broad compound library, but everything in the library was FDA approved in humans. And so we chose that library in that case because we hope that what we might find could be extrapolated to humans much more quickly because everything was FDA approved. The other compound library that we're testing is a library against molecules or compounds that either activate or inactivate generally G protein coupled receptors, thinking, this is a GPCR, so maybe we'll find modulators that way. But we were also hoping to find GPCR modulators beyond GPR126 because they're so druggable. That's how we went about it.
Great. So are there drug companies that are interested in, for example, the Stockel as a drug candidate?
Yeah, I think the Stockel as a drug candidate is so yes, the short answer is yes. This is a really attractive potential drug candidate. So I wasn't able to get into all the biology of adhesion GPCRs. There are actually 33 of them in humans. There's a lot of them, and they do beyond myelination. They actually do a lot of really important things in the nervous system and the immune system. Every adhesion GPCR seems to have its own Stockel, and that Stockel is specific only to its own receptor. And so this is a really, really specific activator of its given GPCR, and so that's exactly what you want in a drug, right? You don't want it to have a lot of off-target effects. You only want it to target the receptor that you're interested in. So yes, there's a lot of interest.
We have a question. So GPR126 is a protein on the glial cells themselves or on neurons that promotes interaction with the myelinating glial cells?
Great question. We know that it's definitely on the glial cells. GPR126 is on the glial cells, and we know that actually from our work in mouse. In mouse, we've been able to knock out GPR126 not just globally, but in specific cell types. And when we knock out GPR126 specifically in the myelinating glia, this has the same phenotype, the same myelin defect as when it's lost globally. And so we think, at least in this case, it's required in the myelinating glia. However, other adhesion GPCRs are definitely expressed in neurons. And in another example in the lab, we're actually getting, this one is called GPR56. Again, the name doesn't really matter, but when we knock it out in glia versus knock it out in neurons, we're actually getting slightly different phenotypes that when put together are what we observe in the global mutant. And so for 126 myelinating glia, but for others, probably both.
Another question is there a link between myelin and neuropsychiatric disease?
Yeah, that's a great question. So, so the short answer is yes, there is. There is a link. And so you may have heard about something called a GWAS study. So these are genome-wide association studies. And when, so you take an affected group of patients versus a non-affected group of patients, and you see which genes are most tightly linked to the disease state. And genes that are known to be involved in myelination are actually very commonly highly linked to diseases like schizophrenia, bipolar disorder, and additionally to autism, actually. And so this might be something that the zebrafish is really useful for, taking particular, you know, I don't think that we would necessarily model a schizophrenic zebrafish, but we could certainly make these precise mutations and see how that affects the basic biology of these cells.
We have a question. Why is neocortex unmyelinated? I think that's partially true, right? Like there's a bit of myelin in there, but just not a ton.
Yeah, so it's not only is there a bit of myelin, but not a ton, but it looks very different than the textbook, right? So that image I showed you, excuse me, of myelin really uniformly patterned along the nerve, it's actually not that case at all. In the cortex, myelin is quite intermittent and very patchy. So sometimes you'll have really long segments of myelin, really short segments of myelin, or no myelin at all. Why is it like that? I think, you know, putting myelin in the sort of these basic questions we talked about at the beginning, putting myelin at the right spot at the right time. Imagine you have a signal starts here and another signal that starts here, and you need them both to get to this point at the same time. This is really long and this is really short. You know, modulating exactly the length of myelin along this axon versus that axon might be a way to get the signal to the same spot at the same time. And so particularly in the cortex, you know, being able to put it down sometimes but not always is probably really important for circuit prep for higher order circuit function, would be my guess.
And is that why a juggler or a new pianist might be growing new myelin to sort of make those connections better or worse?
I think that's the thought. Yeah, that's the prevailing model in the field, but has not been directly tested.
Since you mentioned differences in how the myelin looks, one person asked, are there differences in myelin biology or the myelin defects, for instance, that you see in different types of MS?
Are there differences in the types of defects that are, so like, does that mean like gray matter versus white matter, or I'm not?
Well, I think of the different types of MS, is it relapsing remitting versus chronic progressive?
I see.
And then do you see like very different things in the myelin biology, or is the relapsing remitting sort of an earlier stage that just gets worse?
Yeah, that is a really good question. I think that's slightly difficult to answer, in part because, you know, when you're looking at a T2-weighted MRI, for example, the resolution is certainly not, you know, what we can get in animal models. And
just so everyone's aware, that's like That's like on the millimeter or less scale, right? So it's a teeny tiny box that you're looking at in the brain. You can't really resolve in a human living brain what's going on.
Right. So, right, in a living brain, it's very difficult to resolve with good resolution exactly what the pathologies are. And sort of at end state, I think, by that point it's quite progressed, and the defects do seem uh, somewhat uniform by that point. I think this is animal models have their strengths and weaknesses, but being able to look at the cells at the different stages of progression in animal models is somewhat helpful here. Um, yeah, I'm not sure. I think, every sort of attack or every plaque had its own starting point and maybe a little bit different. But I do think that, if the goal is to stimulate remyelination, you'll need to understand the microenvironment, and also you should still be able to drive myelination intrinsically with the right stimulus, I would think, if that makes sense.
Yeah. Someone just asked a very thoughtful question, I think, is have you looked for serum contents that reflect glial evidence of disease specificity or other things that would imply disease specificity?
We have not done that. I assume that means in the zebrafish. We have not done that, but that's a really interesting idea. And yeah, one that we've been toying around with in the lab, both going from serum into our animal models, as well as looking more broadly at what happens when the myelin goes wrong.
Yeah. And we actually heard some talks yesterday on how individual patients can actually generate very specific and individual antibodies to attack their own myelin. So there's a really deep and interesting biology there. Yeah.
Definitely.
Why is there more MS in the Pacific Northwest?
Yeah, that's an interesting question. So my understanding is that the prevalence of MS is about threefold higher in the Pacific Northwest, and actually all along the 49th parallel. And so there are many clusters of MS on the 49th parallel. Some thoughts as to why that might be. There's been some thought that maybe this has to do with the distance from the equator, and therefore the amount of sunlight, and therefore vitamin D. I don't think there's any good evidence that vitamin D is that's unclear. If just I don't think a vitamin D supplement is enough. The other thought is that perhaps the ethnic groups that live along the 49th parallel, so in particular Scandinavia and Scandinavian descendants, may have some genetic predisposition to MS that we don't fully understand. There's certainly an environmental component and a genetic component to MS, and again, we don't fully understand either.
One other question from the group was, can you use zebrafish to look at known human mutations that are based on repeat diseases rather than point mutations to try to understand that disease?
Yep, you certainly could. And so, I mentioned that we could make precise mutations as long as that stretch of DNA is conserved. But even if it's not, one thing I didn't really get into, but because the fish develop externally and in the water away from mom, we can inject anything we want. You saw that two-celled embryo turn into an animal. When it's still in the single cell or 2 or 4 cell stage, we can inject anything we want into the yolk of the fish, and then it gets incorporated into the whole animal. So it's really easy to do overexpression. So yeah, it's like polyglutamine repeats or things like that. We can simply either overexpress globally or use specific promoters of our cell types of interest to drive particular repeat diseases. That would be quite straightforward to do in the fish. You might be muted.
Sorry.
Okay.
I muted myself so I wouldn't distract. A lot of the power seems to be in zebrafish the ability to do these genetic screens. And could you just talk a little bit about how much work those are? Because, you know, some in the audience might be thinking about how could philanthropy really help the lab to sort of power things forward? It seems to me like that would
Yeah,
be one great way to do that.
yeah. I mean, I will say that with the robot you bought us, it is a lot faster than it would have been without the robot. So thank you. So in that cartoon, I showed you that we mutagenize and then we screen the great grandchildren. And so basically, you're limited by how long a generation time takes. It takes about 6 weeks to go. We hit the fish with mutagen once a week for 6 weeks, let them recover for a while, mate them. It takes, 6 weeks to 3 months for that generation to grow up again. So once you do the mutagenesis, you're about a year away from screening. And then you can screen really for as long as you want. This is always the struggle of a genetic screen, actually. How far do we push it and keep screening and keep screening? And when do we stop screening and start to analyze the mutants that we have in hand? The last screen that we did back when I was at WashU, I think we screened, it took about a year to get all the lines ready, and then we screened for about a year. And then now we have all these mutants that we're following up.
And that was how many people working on that screen?
That was a collaborative screen between 3 labs, so there were a lot of us. But if it was just going to be my lab, about 5 people.
So that the point there I'm trying to make as the guy who has to try to help find ways to support your lab is these aren't cheap, but they can lead to really amazing discoveries like GPR 126, which was one of the first and most exciting things to come out of the first screen for myelin mutants. So I don't think you can overemphasize the importance of the zebrafish in that area.
Yeah, and also with the power of the screens, I mean, just to point out, there's a term in genetics called saturation, and that's when you keep getting the same mutations over and over and over in your screen. We're not to that point at all. Every time we do a new screen, we find completely new genes. And so what that means to me is that there's a lot of discoveries left to make, that using this tried and true approach, there's still a lot of low-hanging fruit there.
So there are gonna probably be some weaknesses to the system. No system is perfect. What are those, and how do you try to overcome them?
Yeah, so I think some of the weaknesses of the fish are we tend to do all of our studies in very early development. And part of that is really just logistical reasons. So the fish, the baby, the larvae have a yolk sac for the first couple of days of development. You don't have to feed them. If you're not feeding them, they're not excreting waste. If they're not excreting waste, you can just keep them in a petri dish and you don't have to put them on the system with the tanks. So it's a lot easier to do your experiments in a petri dish versus, using a net to get your fish out. And once they get a little bit older, they're not transparent anymore. And so we tend to do, with some exceptions, but we tend to do almost all of our studies in the larvae. So I think a caveat of that, even though it's really fast, we can do really nice live imaging, we can do really high throughput genetics and chemical screens. It is still a larva, right? And so if you take a newborn mouse pup and transect its heart, can actually regenerate its heart, right? So baby animals are better at repair and regeneration than an adult animal. And so we probably are looking at an optimized system for repair and regeneration. I mean, in some ways that's a strength, right? Figure out how the best case scenario works and see if you can harness it. But in terms of thinking about, someone in their 30s or 40s or 50s, a 5-day-old fish is much more like a little baby than a mature adult. So how do we circumvent that? Well, fish is a fantastic model that my lab uses, but it's actually only about half of what my lab does. We also work on mouse models, and so of course, a mouse is not a human either, but it is a mammal, and there are much more established aging disease paradigms, demyelination, remyelination paradigms, as well as slightly more convenient ways of doing cell-specific loss of function analysis. And so when we find that the fish has reached the limit of its utility, we tend to take our questions to the mouse models.
So you do discovery in the fish, and then you sort of march up the evolutionary ladder. Have there been is there anything you found so far that hasn't worked the same way in a zebrafish when you compared it to a mouse or a human? That's sort of a loaded question. I know the answer is probably
Yeah.
not.
No, the answer is probably not. I mean, there's subtle differences for sure, that are interesting biology, but anything that's come from gene discovery in fish, we've found functions in mammalian systems.
Mhm. Well, terrific. I think that in the interest of time, so people can move on with their days, we should stop there. But let me thank Kelly again for a wonderful presentation. That was really terrific and interesting. Let me thank Kate Stout for organizing this whole thing and all of the support for getting this out to all the rest of you. And thank you to all the attendees for sharing this time and asking your good questions. And again, please reach out if you have any further questions to either Kate or, as Kelly mentioned, you could reach out to her directly. So let's say thanks again, Kelly. Excellent talk. And goodbye.
Thanks so much, everyone. Thanks for your great questions. Have a good day.
Bye.